most of the critters that you know about, plants, animals, fungi, and protozoans, they have the eukaryotic cell system, but we still carry in them on our bodies a lot of prokaryotes. In fact, the number of prokaryotic cells on our body, in our body, probably outnumbers the number of actual eukaryotic cells that compose our cells. All right? Okay. Now, I'm not going to say, most of this lecture is going to concentrate on eukaryotes because eukaryotic structure is pretty complicated. And whereas prokaryotes are biochemically complicated, all living things are complicated, but nonetheless, the basic structure that you can observe through a microscope is not so profound. So all bacteria, okay, bacteria and archaeans, they of course have a plasma membrane, a cell membrane. Plasma membrane, cell membrane, same thing. I prefer to call it plasma membrane, but I would accept cell membrane any day. Same thing. Okay, phospholipid bilayer, and I'll have more to talk about that later on. What they don't have inside of their cytoplasm, the interior part there, is the space is divided up by other internal membranes. And this is really what's big about eukaryotes. The DNA is much, the whole genome is on one single DNA molecule, circular in shape, not a whole lot of proteins associated with it. It's still very big, but it is sort of wound up and fitted to the cytoplasm. It's not enclosed in any sort of nucleus. That is, the area around it is called a nucleoid. I usually don't talk about that because people get confused and then end up saying the prokaryotes have a nucleus. They don't, okay? This is something else. And furthermore, on the outside of the cell wall, most of these also are outside of the plasma membrane. They also have a rigid cell wall, and I'll have more to talk about that when I get later in the lecture. Other than that, they don't have a whole lot. So some bacteria would have one or more long thread-like things that are called flagella, which I'll get into here in a minute, and other shorter thread-like structures that are called pili. Okay, the word's right there. Okay, so I'm not going to talk too much more about them, but I do want to talk about some of the cell wall structure. Okay, first of all, let me say something about the word flagella. We will also see that eukaryotic cells have flagella as well. However, in biology, we frequently have words that some of the words were referring to the same thing. In other words, they're homologous. But other usage is analogous. Okay, I'll put those words up here. Okay, what I mean by homologous is that the structure is pretty much very, very similar, and they have the same evolutionary origin. Okay, for example, your hand and the front paw of your pet dog or cat, they are homologous. Okay, they have very similar structure and same evolutionary origin. But for the economy of words, we sometimes use the same word in an analogous way. Okay, that means that they have somewhat similar functions, but totally different structures, or very vastly different structures, and they have separate evolutionary origins. So flagella in bacteria and eukaryotic flagella, they are vastly different, and they evolved independently of each other. Okay, so that word, and we'll see that in physiology too. We'll find a lot of things that are called gills, okay, that are analogous structures between different groups of animals, but they have different evolutionary origins, needing different structures. All right, so what is the difference here? We'll look at what flagella are in eukaryotes a little bit later, but this is a solid rod, okay, in prokaryotes. It's a solid rod made up of a special type of protein called flagellin. Okay, so this is a polymer of flagellin. Flagellin is a protein. Okay, so we can have higher order polymerization. All right, because earlier I was talking about for nucleic acids, carbohydrates, and proteins, we were talking about polymerization of basic building blocks to make the macromolecule, but we can also have macromolecules, repeated units, and a polymerization of much larger structure. And that's exactly what we have here with this flagella. Okay, and the way it's a solid rod, not very flexible. Sometimes it's sort of coiled, spiral, but the way it is moved is what is embedded in the cell wall and the plasma membrane is a complex structure that I'm just going to call a wheel protein. And it's this wheel protein is made up of about somewhere around 24, 26 separate proteins. And it's a good example of what I was calling a supramolecular structure. Okay? And what happens is that this wheel protein, I should have a picture of it. Come on, where's my picture? There it is. Okay, this is an artist's rendition of it. This is the inner membrane, that's the outer membrane. I'll get to outer membrane in a moment, that's the cell wall. But this protein, this wheel protein, it can rotate. And when it rotates, then that flagellum will rotate too. So it isn't the case that there's the force that's moving the flagella is within the flagellum, it is within the wheel protein, okay? Now, how is this done? Sort of jumping ahead, but since I'm here, might as well. So the bacterium we'll get to this a little bit later in the next lecture about transport processes across plasma membranes, but there is a hydrogen pump. It requires energy, okay? But the hydrogen ions are pumped outside, so we build up a big concentration of hydrogen there, and the only way, so we have a gradient across here, high concentration, low concentration. The hydrogen ions want to come back in, but the only pathway that they have to get in is through the wheel protein. And that creates a torque that causes this torque force that causes it to rotate. Okay, and that's how the flagellum works. When we get to eukaryotes, we see that the flagellum is quite different. Okay, another structure here that I want to talk about for bacteria, and this is mostly for eubacteria, is that their cell wall architecture can be divided into two different categories, something that is called gram-positive and gram-negative. We'll have a lab later in the semester where we'll see what is the distinction between gram-positive, gram-negative. Gram-positive is a type of stain, or gram-staining is a type of stain that was developed long, long ago by a person named Graham. And when you stain a slide that has gram-positive bacteria on it, the bacteria turn out to be purple. And if you stain it with this gram staining technique, gram-negative, they turn out sort of pink and red. Okay, so they turn out different colors, and this is the way in which we can determine whether it's gram-positive, gram-negative. But that staining technique goes back to differences in their cell wall architecture. All right? So what is this distinction here? Easiest is gram-positive. So outside of the plasma membrane, remember the plasma membrane, biological membrane, all biological membranes have a basic structure of a phospholipid bilayer. Okay? There's other things embedded there, proteins, and maybe some carbohydrates on the outside, but the basic structure is still phospholipid bilayer. So the plasma membrane. Outside is a cell wall, and it's a relatively thick cell wall. And it's made out of a structural polysaccharide called peptidoglycan. Okay, so this is a structural carbohydrate polysaccharide. And it's a little bit more complicated than just that because it also has short amines. Okay, when I say short amines, like 3 amino acids or so that link across these fibers. Okay, but for our purposes, that's the cell wall peptidoglycan, and that's all. What's different with the cell wall architecture of a gram-negative is that yes, they have a plasma membrane. Okay, all living cells have a plasma membrane. They have a thin cell wall of peptidoglycan, and they have a second phospholipid bilayer outside of the cell wall. They have 2 membranes, all right? This is the cell membrane. The outer one is called a lipopolysaccharide layer. Okay, polysaccharide layer, because on the outside it is coated with oligosaccharides. Remember oligosaccharides? Okay, so these particular oligosaccharides are somewhat forked. Okay, so they're branched, and most of them are either attached to proteins that happen to be in the plasma membrane or that lipid polysaccharide layer, or to some of the lipids themselves. So it's basically sugar coated on the outside, and so it has that second membrane. So the differences here between gram-positive, gram-negative. Gram-positive has only one membrane there, and it has a thick peptidoglycan cell wall. Gram-negative, they have a plasma membrane, and then a thin peptidoglycan cell wall, and then outside of that, another membrane that is called a lipopolysaccharide layer. In addition, let me mention this too, because it's not here. Yeah, it is too. All right. I mentioned a beta barrel structure when I'm talking about secondary structure. It's a extensive beta barrel, beta pleated sheet that folds back on itself and is embedded in a membrane, forming a pore. This type of pore is only found in this lipopolysaccharide layer, plus some other things I'll talk about later. This is very significant. This is called a porin. Okay, so this porin, it's only in the bacteria in gram-negative, and it's in the lipopolysaccharide layer, and that's going to come back to haunt you a little bit later. Right? So this porin, it's a way in which things can move in and out. Okay, but still they have control over what can come in and come out. So it's not just passive and promiscuous. All right? Okay. So this is all I'm going to say about bacteria. We'll probably add to it a little bit later, but I always do. But right now, let's concentrate on eukaryotes. The two major types of eukaryotes that we're mostly familiar with are, of course, plants and animals. Yes?
So the beer has, what do you call it, the beta barrel? Sure.
The gram-negative.
Gram.
Only gram-negative have, and it's in the outer membrane. Now, this porin structure is not the same thing as a pore. Yes, it has a pore, but I already mentioned other proteins that form pores. We're not talking about one and the same thing. Porin is a special structure. Okay? All right, so there's a lot of internal membranes that are in animal and plant cells. This is color-coded, because actually when you look at this through a microscope, it is clear as water. Okay, we usually have to put some sort of stains on there. This is color-coded just to let you see what it's like. And even plants will have many of the very same organelles I'm going to talk about. However, there is a few differences in organelles between plants and animals. So these internal membranes are called organelles. Okay, and I'm going to go through all these organelles. What's unique here about eukaryotic structure is that the interior cytoplasm is divided up into rooms, separate spaces, compartments. In bacteria and archaeans, it's just one cytoplasm. Okay, but here with the phospholipid bilayers, that separates things into different compartments. Some of the typical organelles that are membrane bound. Okay, when I say membrane bound organelles, their outer boundary is a phospholipid bilayer. Okay, and separating them from the rest of the cell. All right. So I'm going to talk about these. These are all common to both plants and animals, fungi, algae, etc. There are some other organelles that are very specialized that we're not going to cover here. A nucleus, something called the endoplasmic reticulum. Okay, I just have it abbreviated up here, ER. You'll see the whole word when we get to it. Endoplasmic reticulum, something called the Golgi, lysosomes, vacuoles, peroxisomes, mitochondria, chloroplasts. Okay, these are membrane-bound organelles and very common all throughout the eukaryotes. So these are internal membrane systems. So they're within the cytoplasm, and as I said, they partition the organelle into different compartments. And there's some things that can happen here. Also, something about it is that the lipids that make up the phospholipid bilayer are not necessarily the same from one organelle to another. And so they can differ, their membranes can differ in terms of their lipid composition, and also in their protein composition. And in fact, it is primarily the differences in their protein composition, both in the membranes and in the internal space, that give these organelles their functionality. Okay? Much of that is due to the proteins. Some cases there are some lipids that are here too. Now, what is the advantage of this? Well, one, it can isolate incompatible reactions, which the bacteria have evolved ways to do this, but nonetheless, this is a good and easy way to do it. So I talked about hydrolysis and dehydration reactions, okay? Those are going in opposite directions. And if they're all happening in the same space, they can compete with each other. So you'd be putting something together and tearing it apart simultaneously. But if you put certain types of reactions within an organelle and others reactions, competing reactions within a different compartment, they are separated. And there's no possibility of interference. Okay? That's one of the advantages here. Also, some chemical reactions require special physiological conditions. And if you maintain those physiological conditions throughout the whole cell, then certain reactions would be favorable, certain ones would not. They'd be inhibited. And so we can create an organelle and specialize the internal environment for a particular types of reactions, creating a space that might have a lower pH or a higher pH or something like that compared to the rest of the cell that would be more favorable. Also, it's a problem with resources, okay? If we have a certain chemical reaction that's vital, that has to take place, the reactants have to be at a certain minimum concentration, okay? Because with these chemical reactions, you have the enzymes and the reactants, they have to collide, all right? And if it's too dilute, that's going to be way too slow. So we have to have things concentrated sufficiently that we have enough collisions that the reaction can proceed. And so if we have a big cell, eukaryotes is a relatively big cell here, and we will have to have all the reactions all throughout the cell at that high level. Not only for this reaction, but for all the reactions. You'll have to maintain real high concentrations, which would be metabolically expensive and probably impossible with something this high. So instead of doing that, put it inside of a little organelle. You can maintain just that high concentration there, just there, not universally throughout the cell, just there. And this will be sufficient enough to provide for all the products that is necessary for the whole cell. Okay, so that's what I mean by localized concentrations versus global concentrations. We can concentrate things in one compartment such that the reactions can go on, all right? So that's an advantage of having this type of organization. Okay, so I'm going to go through some of these. In fact, I have something back here. Seems like I'm missing something. All right, no, I'll go ahead and talk about the first one, the plasma membrane. Early on, the only membrane that we could get to study was plasma membrane because it's on the outside. Okay, there weren't techniques very well to get to some of the membranes on the inside. Over the last 50 years, biochemical and cellular biology techniques have really advanced that yes, we can get into those membranes on the inside and investigate what they're like. But nonetheless, what we learned about plasma membranes pretty much applies to all the other biological membranes on the inside as well. So, except for this here forms the boundary of the cell, whereas internal membranes form the boundary of those organelles, all right? So some of the things here is, of course, phospholipid bilayer, as its principal structure, 2 layers of phospholipids facing opposite each other, with the hydrophilic heads facing inward and outward, and in the middle is the hydrophobic tails, but it's also what's embedded in it is protein. In fact, most membranes, about 50% of the mass for the plasma membrane, this is sort of an average about 50% of the mass of the plasma membrane is phospholipid, and about 50% is protein. Proteins is what gives function, most of the time. So what it does, it controls traffic in and out, okay? So any phospholipid bilayer also has a principle of being semi-permeable. So if we didn't have proteins, we just had a phospholipid bilayer, then that would be semi-permeable. What it means is that certain types of molecules can easily pass through it from one side to the other, but it opposes the passage of other types of molecules. And so it isn't perfectly permeable, it is permeable only to certain things, and it's impermeable for others, okay? All right, and also this is in, the plasma membrane is involved with cell communication. Communication between cells, communication in other parts of other individuals, if we're talking about single-celled organisms, but it's some of its function. Okay, yeah, this was a slide I was expecting. Okay, the first groups of membranes I'm going to talk about here, organelles, they belong to something that's called the endomembrane system. They are organelles that sometimes are in physical contact, or if they're not in physical contact, there is a lot of exchange of materials between them. And so there's a lot of traffic moving between them. There are other organelles I'll talk about a little bit later that do not belong, still membrane-bound organelles, but they don't belong to the endomembrane system. There's a lot of movement between these organelles. So if they're not in physical contact, a lot of times materials are moving from one to another by means of what are called transport vesicles. These are still membrane-bound structures, but they will bubble out from the membrane of one organelle with products on the inside, form a bubble, sort of like a blister, and then they'll bud off. And then they're transported over to another organelle where that bubble will fuse with that membrane, dumping its contents into the interior of the other organelle. And so there's a lot of exchange and movement here between these particular organelles, okay? So this endomembrane system includes the plasma membrane, nucleus, endoplasmic reticulum, Golgi, lysosomes, vacuoles, okay? Put that back up, sorry about that. These are members of the endomembrane system, okay? And now, endo means inside. Plasma membrane isn't inside, but it is still part of the endomembrane system because there's a lot of interchange between the plasma membrane and these other organelles, okay? All right, okay, so first of all, nucleus. Just talk about the big parts of it. This is what contains in eukaryotes most of the cell's DNA. Notice I said most. Most doesn't mean all. Most does not mean all, okay? Most of the DNA happens to be inside the nucleus, which is technic. This is usually one of the largest organelles you can see in the cell. Even with a light microscope, with staining, you'll be able to see the nucleus, okay? And you'll have a chance in the lab later on to be able to do so. So it's about, on average, about 5 micrometers across. And as far as the structure, it is surrounded by a phospholipid bilayer that is called, actually 2 phospholipid bilayers, called the nuclear envelope. So this is not just one phospholipid bilayer, it's 2, okay? Looks like I can't bring my pointer with me. Okay, so if you can look up here at this figure, can't point anymore, but up there, there's an outer phospholipid bilayer and an inner phospholipid bilayer. So in between the two, there's a narrow fluid-filled space. So in between the two, there's a narrow fluid-filled space. Okay, and so that is the nuclear envelope. And of course, the nuclear envelope has, just like any membrane, it has proteins embedded in it that give the nucleus their functionality. The shape of the nucleus is maintained by a meshwork, sort of a network of linear proteins that form what's called the nuclear lamina. And this is coding the interface of the innermost nuclear membrane.
okay? So it's just right on the inside. Furthermore, a lot of chromosomes happen to be attached to the nuclear envelope or nuclear lambda, okay? Now, things have to move into and out of the nucleus, and therefore this is perforated. Both of the ligers are perforated by protein complexes that are called nuclear core complexes. And so these are about the size of 100 nanometers. This, once again, is another example of a supramolecular structure because it's not just one protein. Okay, it's lots of proteins, and it does form a pore. Okay, an aqueous pore. It's not a porin. Okay, porin and the lipopolysaccharide layer is one protein that is in a beta barrel shape, but this here consists of lots of proteins. Okay, lots of proteins, and it is so traffic can move in and out of the nucleus. Now this is selective, so there's special types of proteins that just collectively called escort proteins that are involved in moving things in and out of the cell. Now this is a broad term because there's many of these different types of proteins that give special names. Okay? All right. Now within the nucleus, this is not another organelle. It is just a dense space, okay, but it has a special function. This is called a nucleolus, and so it's not membrane bound, okay, so it's within the nucleoplasm. Okay, nucleoplasm, that is the name of the fluid that's inside of the nucleus. Nucleoplasm, and so this is in the nucleoplasm, and it is an area of dense material. It has a lot of nucleic acid and proteins, okay? And what is special about it is the site of synthesis for ribosomal RNA. I mentioned that there's going to be several different types of RNA. So this is called ribosomal RNA, and that plus proteins are put together to form subunits of another structure that I'll talk about soon called ribosomes. Okay, so this is not where ribosomes are made in the cell, it's where their subunits are made. And their subunits consist of ribosomal RNA and a horde of proteins, right? So this happens there, okay? Furthermore, where the nucleolus forms inside of the cell is along one or two chromosomes. Chromosomes at this time, the DNA is really relaxed, and so it's really spread out. But there's a part of the chromosomes, could be on one chromosome, could be on two, that is called the nucleolar organizer. And that nucleolar organizer is where it forms. Okay, that's the nucleus. Here we have one chromosome. Here it might have another chromosome. They come close together right there. And what we have here, what constitutes the nucleolar organizer, is that it has special units of DNA genes that encode ribosomal RNA. Okay, I'll get to it a little bit later, but I mentioned that proteins, of course, differ by the amino acid sequence. Okay? It's a gene that codes the information for the amino acid sequence of a particular polypeptide, as we'll get to a little bit later. But also with nucleic acids, although the alphabet is shorter, there's 20 amino acids for proteins. For nucleic acids, there's 4 letters to the alphabet, okay? But they can be put together in a very specific sequence, and their sequence does determine their function. And that has to be specified somehow, and that's what these genes do. They contain the information for the exact order of bases within a ribosomal RNA. So this is where they are being copied, and the oxyribonucleic nucleotides are being produced in this area, linked together with proteins. And because we have this high attraction of all that material being produced there, it creates that big space that we call a nucleosome, okay? Or nucleolus, sorry. So the nucleolus is a space where that density is. The nucleolar organizer is part of the chromosome where that nucleolus happens to form. Okay? All right. All right. So here's a picture from another. You don't have to memorize all this stuff. The only thing about this one is the better picture that's in your textbook, sort of showing a little bit better what's going on here. This is the cytosol, and this is the outer membrane. This is the inner membrane. Here we have that fluid-filled space, and there's all sorts of proteins that are still associated with this. This here is the nuclear pore complex, even though it shows it like a barrel, that is actually consists of a lot of different protein, separate polypeptides. Many of them are sort of globular in shape, so that they would form sort of a hook together like that. And if you're looking at it from up here, you sort of see all these proteins embedded. A lot of their R chains sticking into the lumen, and that also plays a role in how the escorts, which escorts can get through it. Okay, so these are the major features of the nucleus. There's more things, but that's enough for now. Okay, now 2 words that are often used interchangeably. We'll come across things like that. I've already mentioned one conflict or special case of polypeptide versus protein. Okay. There's a similar story here with 2 words, cytoplasm and cytosol. So technically speaking, cytoplasm is all of the space and its contents between the plasma membrane and the nuclear membrane. Okay, so that's all the space, and it contains the organelles in eukaryotes. Okay, that's part of the cytoplasm, and in between the organelles is sort of a viscous fluid. Fluid area is called cytosol. Okay, so really there is a difference here between cytosol and cytoplasm in eukaryotes. There's no distinction really when it comes to prokaryotes because they don't have any membrane-bound organelles on the inside. So you can call that cytosol or you can call it cytoplasm. Here there is a little bit of distinction. Okay, but a lot of times in the textbook and even in my usage, sometimes I'll be jumping back and forth all the time. Because if I say it goes into the cytoplasm, I might, cytosol is part of the cytoplasm, but I might specifically mention cytosol, which means it's not going into any organelle. It's going to be found in fluid-filled space between the organelles. All right? Okay, now I'm going to diverge here because I was talking the whole theme here, but story, endomembrane system. Okay, but in order to understand one of my next organelles I'm going to talk about, I got to diverge and talk about ribosomes. Ribosomes are not part of the endomembrane system directly, and they're not membrane-bound organelles. However, I have to say something about them. They do not consist of a phospholipid bilayer. What they're important for is that this is the area of this is the agency of protein synthesis. This is where amino acids are put together to form polypeptides, right? And they consist of many proteins, depending on what organism we're talking about. Ribosomes in bacteria consist of fewer proteins than in eukaryotes. Now notice here, this is an organelle. And when I went over bacteria, difference between prokaryotic cells and eukaryotic cells, I said that they didn't have, eukaryotes did not, or prokaryotes did not have many membrane-bound organelles. I did not say that they didn't have organelles. You really, when I speak, you need to really catch every word, okay? Because this is an organelle, and bacteria do have it, okay? Because they do make proteins, right? They have, so it consists of proteins, lots of proteins, okay? And ribosomal RNA, okay? In fact, usually within the ribosome, there's 3 different types of ribosomal RNAs, all right? And I'm not going to go any more into that, but that's their structure. So you find them, thousands of them scattered throughout the cytoplasm of the cell. These can be free or bound. Now, what I mean by free or bound, if it's a free ribosome, it is in the cytosol, and it's functioning in the cytosol. If it's bound, it's bound to the outer membrane of the endoplasmic reticulum, which I'll talk about shortly, or the outer nuclear membrane. And so there's nothing different about a bound ribosome versus a free ribosome except their location and the general class of proteins that they're producing. That's all. So the one and the same ribosome could be free at one moment, then attached to the endoplasmic reticulum. It's now a bound ribosome. And then a little bit later, it could be a free ribosome again. Okay? So there's nothing structurally different about them. What is different about them is their location and the general class of proteins that they happen to be making. Okay? All right. Okay. Now, prokaryotes have ribosomes, but already there's differences between prokaryotic ribosomes and eukaryotic ribosomes. So some of the differences here is in their size. Now, a dalton is the mass of a hydrogen atom. Okay, you don't have to know these numbers. The whole point I'm bringing out here is that eukaryotic ribosomes are bigger. Okay, they're bigger, almost 45% bigger. Okay, larger than prokaryotic ones. Eukaryotic ones are lower. That's because they have more protein. Okay, they have some proteins that aren't found in eukaryotes or prokaryotes. Incidentally, there is homology between some of these proteins. We can recognize that this is a very ancient structure. Okay. Furthermore, prokaryotic ribosomes can be inhibited by tetracycline, streptomycin, and similar types of antibiotics. Antibiotics kill bacteria in a variety of ways. So we have different classes of antibiotics. This doesn't, something like penicillin doesn't do this. Okay, this is not how penicillin kills. This is tetracycline and streptomycin. They kill bacteria by inhibiting their ribosomes. Okay, if the bacterium can't make its proteins, it'll die. Okay, so that's a favorite thing. All right, and this, these two do not harm eukaryotic ribosomes. So that points to must be some structural difference. We're not going to go into details about what that structural difference is, but there must be some structural difference whereby prokaryotic ribosomes are inhibited by these two antibiotics. Okay, so this is a figure from your textbook showing cartoon of a ribosome. They consist of two subunits: a large subunit, small subunit. Okay, and these are normally not attached to each other except for when they're engaged in protein synthesis. So what is made in the nucleus, in the nucleolus, is the subunits. Okay, this consists of a large number of proteins and some ribosomal RNA. This is also proteins and ribosomal RNA, different ribosomal RNAs, and they only, they're assembled in the nucleolus, inside the nucleus, and transported out. So those little dots up there, on the upper figure, those are ribosomes. Okay? Now, you have to have ribosomes to make a ribosome. So when a cell divides, some of the ribosomes have to go to all the two daughter cells. If you destroy all the ribosomes, then it won't be able to make ribosomes, because it's a ribosome that makes riboproteins. Ribosome consists of some proteins, and where the ribosomes function is in the cytosol, not in the nucleus. They're in the cytosol. So here on the cytosol, we have a ribosome. It gets instructions, and we'll talk about how it gets instructions later, from the nucleus, from the DNA, as to how to put together the amino acids to make these particular ribosomal proteins. Then these ribosomal proteins have to be transported into the nucleus, okay? Through the nuclear pore complex, okay? It's a too big to fit through anything else. So that's its passageway in. They're brought to the nucleolus. Nucleolus is where the nuclear organizer is. Here I have 3 chromosomes there. These have repeated copies of ribosomal genes, okay? Because we need to make a lot ribosomal protein. The way to do it is to have extra copies there. So here's where we have all the ribosomal RNA that's being manufactured. All that protein is brought here, and then the subunits are put together and then transported back out through the nucleophore complex. So it's the subunits that are made in the nucleolus, then transported out. Then they will go together, large and small subunits, into a functional ribosome when they're engaged in protein synthesis. Okay? All right, so that's the ribosome. Now, why do I bring it up? Because it is associated with the endoplasmic reticulum. Okay, the endoplasmic reticulum is another organelle. It is a maze of tubes and sacs that go all throughout the cell. Okay, and it has one membrane around it, endoplasmic reticulum membrane. And there's two different varieties of this. Something is called smooth ER, because you can use the abbreviation for endoplasmic reticulum as ER, that's fine with me, and a rough ER. Now the internal space, that is the fluid-filled space inside of these sacs and in these tubes, typically it's called a cisternae, but I'm going to use the word lumen. Okay, a lumen is a general word that applies to any interior space in any bag or tube-like structure. For example, all that space in your digestive system, in the stomach and into the small intestine, large intestine, interior space is called a lumen. All right? So any space inside these membranes is also called a lumen. So there's enzymes in there. Now, what the physical difference between smooth and rough ER that can be seen through the electron microscope is whether they have ribosomes bound to the cytosol surface or not. Okay, so smooth ER if there's no ribosomes bound. It is bound or rough ER if it has ribosomes bound to the cytosol surface. This is what makes it look rough through the microscope. It's all those little bumps on it. That's why it's called that. Not only is there a physical difference here, okay, this here is rough ER, bunch of tubes right here, and all those little black dots, those are ribosomes, and they're hooked to the outer cytosol surface. Now, there's also all sorts of enzymes in the lumen, and rough and smooth ER don't only differ by how they look, they also differ in their general functions, all right? So some of the functions of smooth ER this is where the synthesis of lipids takes place. So there's enzymes in the lumen, and they will assemble, for example, cholesterol. If it's in the testes or ovaries, some of those types of cells are loaded with smooth ER, and where some of the function there is to produce hormones such as testosterone and estrogen. Okay, and those enzymes are inside of the smooth ER. It is also a place for carbohydrate metabolism. Some of the oligosaccharides that are produced on the outside of the cell, which I'll get to a little bit later, some of the enzymes there are in the smooth ER, and also glycogen being stored in the liver, that's involved here too. It is also one of the two major detoxification centers of the cell. Okay, and mostly detoxifies, it has a class of enzymes inside that they hydroxylase, attach hydroxyl groups to the toxin. Here, it's mostly taking care of toxins that are hydrophobic. Where in the cell would you expect hydrophobic toxins to accumulate? In general. In the cytosol? Where would they tend to accumulate? The hydrophobic? They would need to go to a hydrophobic space. Where can they go to a hydrophobic space?
In the bilayers, possibly?
In the bilayers, okay? And in fact, some of these toxins, that's exactly why they're toxic, because they're interfering with membrane function. And so what happens if you attach hydroxyl groups to that toxin, which is hydrophobic? It makes them polar. Are they gonna stay in the membrane? No, they'll go out of the membrane. For one thing, it changes the nature of the toxin. Anytime you attach something, some other atom or molecular substance, something, it will change its nature, but it also makes this more polar, so it can come out of the membrane and be easily flushed out of the body. So this is a big class of important proteins that are involved in this. They're called cytochrome P450. So this is a family of different enzymes that are involved in this detoxification process in the smoothie R. Okay, some other functions is that this is a place to sequester calcium ions. Okay, as we'll see a little bit later on, mostly in physiology in next semester, is that calcium has some very important functions. Okay, in muscle contraction and in nerve impulse propagation. And so it has to be controlled. So in order to control muscle contraction, the calcium has to be removed from the cytoplasm and put back into the cytoplasm where the contractile proteins happen to be. And it has to go in cycles, all right? So it is sequestered a lot of times in the smoothie R, okay? And we'll come to that later. The rough ER, this is the site of where ribosomes produce a particular category of proteins. When I talk about the differences between the proteins made by free ribosomes and bound ribosomes, I'm making very, very, very broad generalizations. That is mostly true, but not 100%. This is one of the things we're going to say in biology, is that at this level, we instructors lie to you. Okay, we tell you half-truths, all right? A little bit later on, when you go on, and we hope you forget we told you that, and you'll get more details, all right? But life is so complicated, a lot of the things we say are broad generalizations, okay, because there's always exceptions, all right? So here, this is the site of secretory proteins and membrane proteins, of membrane proteins that will function somewhere in the endomembrane system organelles are made here. It isn't the rough ER that's doing the synthesis, it's the ribosomes, okay? The ribosomes are bound to the outer membrane. They're producing certain types of proteins. What do I mean by secretory proteins? They're proteins that are going to function outside of the cell. So they've got to be dumped outside, all right? And so ribosome attaches to the surface, is making a protein. Usually there's a pore complex there that the polypeptide is fished through the membrane into the lumen, and then the protein folds on the inside. And we'll get to a little bit later how that protein gets from the rough ER to the outside, okay? And so forth. And so all of the endomembrane organelles, they have proteins in their membrane, they have proteins in their lumen. And so most of these proteins are being made by ribosomes, bound ribosomes on the rough ER, okay? And then they get transported somewhere else. And of course, smoothie R is involved in membrane function too, but because of but all membranes, so the lipid part of a membrane, smoothie R, okay? And smoothie R, rough ER are continuous. They're joined together. These things can move over to the rough ER, where then proteins are put in as the final product of the membrane. Okay, because all membranes will have their proteins in there. Okay, they're not just bare phospholipid membranes. All right, so this, in other words here, the ER is a major manufacturing center of the cell. Major manufacturing center of the cell. And all the materials that's made there have to be shipped to where they function, okay? So these products are then shipped through a transport vessel to another organelle called the Golgi, all right? So a transport vessel buds off with the product, and it will fuse to the Golgi, dumping its products there. So the Golgi, it looks like a stack of pita bread. So it has these membranous sacs that are stacked on top of each other, okay? And it has two different sides to it. It has a cis side and a trans side. Now, what is meant by cis? I've mentioned same side, okay? And what do I mean by same side? In this particular context, it is the recipient side. So transport vesicles is the assist side that's facing the rough ER, and transport vesicles butt off of the rough ER. We have a vesicle here, okay, and that blue shows it right there, and it has product, both in its membrane proteins, and in its lumen, proteins that will do something else. And then it fuses with the cis face of that out one sac, dumping its membrane, or dumping its contents into the lumen of that first Golgi compartment. And furthermore, that transport vessel's membrane now becomes part of the membrane of the Golgi, all right, because they fuse together. And then either some of these chambers are connected together so that some materials can move from one to another, but very frequently at the very end, a transport vesicle will butt off, come around, and butt into the next layer. In other words, materials are moving through. They're received on the cis side, and they move through this stack of membranes, and they will exit on the trans side and be taken to their final destination. So there's enzymes, both in the membrane and in the lumen, and some of there's a bunch of metabolic activities that happen here, okay? Some of these will modify phospholipids, okay? Altering them. There may be phospholipids that are brought in here in the membrane that oligosaccharides are attached to their surface, or other changes like that. This also targets the products to their final destination. Where do they need to go? Because what's brought here in that first transport vesicle may contain proteins or products that need to be in the lysosome, others that need to be in the plasma membrane, others that need to go here, others that need to go there. It's all mixed together, but they get sorted out here and separated into, at the end, purified into a vesicle that is going to one destination. So this is more or less the shipping and receiving center of the cell. Everything from the manufacturing center has to go here, and it gets separated. Okay, so here is a probably much better figure than that's in your textbook, because I drew it. Okay, bound ribosome, it's making a polypeptide. Polypeptide folds into its native conformation, and then we have transport vesicle off of the Golgi, and it's now a transport vesicle. Actually, that's the it will fuse then to the Golgi, and as it transverses through all those stacks, it gets modified, sorted, and loaded into, on the trans side, into a transport vesicle that will go to various other destinations. Okay, it could go to the plasma membrane. Some of this can turn around and go right straight back to the ER. It goes that way too. All right. So there's a lot of traffic that's moving through here. Okay, so go back. Okay. Any questions? All right. Okay. All right. Another organelle endomembrane system is a lysosome. Okay. This is they contain hydrolytic enzymes. Okay. All hydrolytic enzymes for all four groups of macromolecules. So, what does a hydrolytic enzyme do? It catalyzes what type of reaction? What type of reaction does a hydrolytic enzyme catalyze? Hydrolysis. And what is hydrolysis? What is hydrolysis? Breaking a linkage with water. Okay, adding water to a linkage. And so, yeah, this has lipases. These are the special ones that catalyze separating fatty acids from phospholipids or from glycerol. Okay, and carbohydrases, variety of them. Okay, for glycosidic linkages, proteases for hydrolyzing peptide linkages, nucleases for nucleic acids and so forth. So this here, the internal lumen is a pH of 5. Okay. Good example of an organelle where a special condition has to be maintained inside it. Okay. These enzymes work in an acid condition. Bring it up to a pH of 7, like in the rest of the cell. It denatures these enzymes, they don't work. Okay. So there's a 40 or more, depends on what organism you're talking about. Lots of enzymes here that break this down. So this is the major digestion center of the cell. Okay. Recycling center. All right. So now where did the membrane come from? It has one phospholipid bilayer, of course. Well, it was first of all put together in the ER and shipped to the Golgi. And then those phospholipids went through the Golgi and were sequestered together for lysosome phospholipids, which could be different than the types of phospholipids that are found in other organelles. And it buds off on the trans side of the Golgi. All right? So it looks like a transport vesicle. All right? Very much looks like similar to a transport vesicle, but it has a special function. So its functions is intercellular digestion. Okay? Things wear out. Other organelles malfunction, and other proteins are no longer needed, or they have been damaged to the point that they're not functioning anymore. But the amino acids that they're composed of can be recycled. You just break them down and reuse them. And so you don't just throw them away. And so that's what's going on here, okay, in part. Also, cells have to take in material from the outside, okay, especially if they're heterotrophs. That can be broken down. Okay, for example, some of your immune cells, macrophages, for example, they're going through your body and they find bacteria, they'll take it, the bacteria digest it, and this is where it's happening, okay, lysosome. All right, so recycling, this is called autophagy. When you see the word phage, phagy, like this, it means phagos, means to eat, okay? Auto doesn't mean it eats automobiles, okay? It means self-eaters, right? It's eating itself. So, and also, lysosomes are involved in programmed cell destruction because during development, especially of a multicellular organism, there are structures that have to be destroyed. Okay, as you, for example, tadpole. Tadpoles, baby frog, they have a long tail, but they have to go through a metamorphic transformation where the tail has to be absorbed. That has a bunch of cells and everything in there. All that material can be used again, it can be recycled, and so it has to be, the cells get destroyed and the materials are reused, okay? So, several things are happening here. This is a cell, and we have lysosomes. Let's look at the autophagy, okay? So, first of all, we have an organelle that I haven't talked about yet, but I will. It's not working anymore. And so, endoplasmic reticulum, an ER-derived membrane, is inserted around it, okay? And then it will fuse with a primary lysosome. Now that contains hydrolytic enzymes. So these two membranes fuse, and now we have a secondary, or autophagosome, it can be called. And this here, all the proteins get broken down to the amino acids, all the complex carbohydrates into monosaccharides, etc. And then they're shipped across so that the cell can reuse it again. Okay, just use the material for something else. But there's also, we'll go up here, taking in materials from the outside. This doesn't have to be a bacterium. Okay, it could be a cell that just has to eat up larger proteins that it finds on the outside of its body. Still nutrients. It's taking in nutrients of some sort, especially if this is a heterotroph. So this, first of all, becomes an endosome. This up here calls it a phagosome. I'm going to call it, this is a more general word. A phagosome is a type of endosome, okay? So it's an endosome. Zone means body, endo going to the inside from the plasma membrane. And that will contain, here it's containing a bacterium, but it could contain some other macromolecule, all right? And it will fuse with a primary lysosome, become a secondary lysosome. All that's going to be broken down by these hydrolytic enzymes, and it's going to be pumped. The material will be released into the cytosol so they can be recycled and used, okay? All right, so these are some of the things. Don't worry about this here. Well, they have endosomes here, good, all right? This here is another type of taking in just only fluid material and smaller particles, but I'm not going to concentrate on that. All right, so that's the role of lysosomes. Vacuoles. This is a sort of a heterogeneous group, you know, because there's many different types of vacuoles, so I'll just mention some of them. There are food vacuoles, okay, where material is being stored or being broken down for being able to use. Contractile vacuoles, and certain protozoans are living in fresh water. Okay, protozoans are living in fresh water. Paramecium. Later in lab, a few weeks down, you're going to play with some live paramecium. I will let you feed them. Okay, you get to watch them under the microscope. All right. So one of the problems with freshwater protozoans, the protozoan single cell, is that they're in fresh water. And the solute concentration in fresh water is very low. Unlike seawater, there's not a whole lot of solutes there. But being a living system, cytosol inside of this organism, it has a high solute concentration. Now, if you didn't learn this in chemistry, you're going to learn it now, something called osmosis. Okay? Osmosis is the movement of water, okay, across a semi-permeable membrane, okay, from an area of low solute concentration to an area of high solute concentration. So inside, we can't make this any diluted, okay, more dilute, because we have to have solutes, proteins, carbohydrates. So inside there is a high concentration, and therefore water has a tendency by osmosis to move in. Okay, now this is a type of diffusion of water. Diffusion of water. But is there something strange about this? I said from low solute concentration to high solute concentration. Is that how diffusion works? Anything in chemistry?
Usually like goes like backwards in the sense like
Say that again?
it goes downwards. Like let's say you have like a lot of heat in one area, the heat will diffuse to a place of low concentration of heat.
So you say I'm telling you, I'm contradicting, I'm wrong here?
No, because water is kind of
That's a pointed question. Are you telling me I'm wrong?
Water is at a high concentration. So
Yeah, because it's a diffusion of water. We're not talking about the diffusion of particles, solids. So where the solute concentration is high, the water concentration is low, because it's displaced by solute, okay? And where the solute concentration is low, the water concentration is high. So out here, this is high H2O. This here is low H2O. So yeah, we have the diffusion of water. But a lot of times we don't measure the concentration of water. So with osmosis, it's easier to talk about the solutes, okay? So it's movement, as long as the solutes can't move, okay, the water is going to be moving from an area of low solute concentration to high solute concentration. Okay, now, why am I diverging on all that? Well, this guy is going, these things do not have cell walls. Plasma membrane. Plasma membranes aren't all that strong. Water keeps coming in, they're going to burst. Okay? But they have a vacuole, contractile vacuole, that takes up the water. And then that vacuole will eventually fuse to the plasma membrane, dumping water out. And this is going 24 hours or 24 hours a day, constantly. It has to constantly pump the water out. Okay, so this is a type of vacuole. Plants, they have a huge central vacuole. Okay, so usually the central vacuole takes up most of the interior, and it's a water-filled thing. and most of the not living, but the metabolically active cytoplasm is around the outside of it. That was one trick that can be made by plants so that the cell size can get larger. Okay, because the metabolically active part does not include all the volume of that vacuole, all right? But there's another different, at this level we have a little bit of difference also between plants and animal cells. Plant cells usually do not have lysosomes. Okay, they have a few, or certain ones might have them, but most plants don't. Instead, the function of lysosomes is fulfilled by the central vacuole in plants. Okay, this is where hydrolytic enzymes happen to be. Okay, so that's in plants. There's other types of vacuoles in plants. Where does the pigments and petal cells of flowers happen to be contained? Usually in a vacuole. Okay, these are light-absorbing features that reflect or transmit different colors of light. Also, some plants are toxic, and so those toxic compounds are also toxic to the biological system of the plant as well. But they're for preventing herbivores and encouraging herbivores not to eat them. Because if they eat them, they get sick. Okay, if it's a smart herbivore, it will leave it alone. But those toxins are in the vacuoles. Okay, they're there where they're not causing any harm to the plant itself. All right, so just the endoplasmic membrane system, a little bit of a review here. Okay, so the major manufacturing center, the ER, that's where things are being made. They're being shipped via transport vessels to the Golgi. This is where they get sorted, modified, redirected, and those transport vessels from the side of the Golgi can be transported to the plasma membrane, either to insert proteins into the plasma membrane or to dump proteins out. Okay? For example, I'll give you a secretory protein. You have cells in your immune system called B cells. They turn into plasma cells. When you have an infection, they are making antibodies. Antibodies works in your blood and body fluids. Okay, they go outside. That's where they work. And so where do they begin? An antibody would begin its life on the rough ER. The antibody is a protein. It's put together by ribosomes on the rough ER. Then it is in the lumen of the rough ER. Then it will be in a transport vessel to the Golgi, transport vessel to the plasma membrane, and when it fuses to the plasma membrane, the internal contents of that transport vessel are folded to the outside. And so it's secreted outside the cell, or other destinations. And even some of these transport vessels can go back to the ER. Okay, so there's a lot of traffic. Really, most of the pictures you see in your book are rather static, and you really need to get in your mind that things are always in motion. Okay, things are not at a standstill. There's a lot of activity going on here. All right, proteins made by free and bound ribosomes. So I said that the ribosome is a ribosome. The differences is where they're found, cytosol or on membrane of the ER, but also in the general broad class of proteins they're made. Okay, this is a broad generalization. Free ribosomes are making proteins that function in the cytosol or in other non-endomembrane system organelles. Okay, as a general rule, it's a broad generalization. They are making proteins that are functioning in the cytosol, or they will be shipped to a non-endomembrane system organelle. All right, that's where they function. Bound ribosomes are making proteins that will operate outside of the cell, or they will operate somewhere within the endomembrane system, either in the membranes of an endomembrane system organelle, or in the lumen of an endomembrane system organelle. So this is generalizations, okay? All right, there are other, this is another figure I got from a different book, because once again showing this vesicle traffic from ER to Golgi, secretory, transport vesicles, dump stuff outside, or a lysosome, okay, and it will take in an endosome, etc. And you see that some of the traffic goes right back to the ER as well. Okay, so there's a lot of movement here to be considered. They're not just objects that are just sitting still. Okay, so those are the endomembrane system organelles, but there's some organelles that are not part of the endomembrane system. And that's what I want to get at right here. So something called a peroxisome. Okay, now peroxisomes are out in the cytoplasm, cytosol, and they bud off. How they originate is that they grow from other peroxisomes. They bud and they rise from there. Their function is twofold. This is oxidation of fatty acids, and it's one of the two places for oxidation of fatty acids. I'm not going to say too much about these fatty acid lipid oxidations, but if you have a fatty acid and oxidize it, you just divide it into two shorter fatty acids. We'll come back to that a little bit later. Okay. It is also a detoxification center. Okay. So we have two major detoxification centers, the SLUDR and peroxisomes, but they function a little bit differently. Okay. What happens here in the peroxisome, there's two different classes of enzymes that are important here. Something that are called oxidases. This is a class. When we say oxidase, we're not talking about a specific oxidase. There's a lot of different specific oxidases. But we have a toxin. One way that it can alter that toxin is to strip off hydrogen from it, okay? And combine that hydrogen with oxygen. So oxygen is needed for peroxisomes. And this, by taking that hydrogen off, it will make a different creature here that may not be toxic, but it produces something that can be real dangerous to the cell, hydrogen peroxide, all right? These have to be taken care of. Okay, that's one way that it can detoxify. In some cases, instead of this being a toxin, this might be a hydrogen donor that is made by the cell to make the hydrogen peroxide, okay? So once again, it's combined with oxygen to make hydrogen peroxide. And then we have another enzyme called catalase that uses the hydrogen peroxide to maybe detoxify another type of toxin, okay? By combining it together, here once again, we're having this reaction, we're removing hydrogen and producing two oxygens, okay? And that changes the nature of the toxin. So these two equations, that could be a toxin, or it could be a hydrogen donor that the cell is making, that the peroxisome is using to make hydrogen peroxide. And it uses the hydrogen peroxide to detoxify. But if we have too much hydrogen peroxide, it can be a problem. This is very reactive and it can tear cells. So the catalase will also do this. So this here reaction with catalase that I have up here is a detoxification. But this here is just getting rid of our excess peroxide. So two hydrogen peroxides with catalase, two oxygen, or one oxygen and two water molecules. So this is also a major reaction that it has. Most of what catalase does is a lot of times this. Okay. All right. So that's peroxisomes. Then there's another set of organelles that are energy converting organelles. One of these is called mitochondria. Okay. So this is the site of where most of the steps of cellular respiration takes place. This is where your food is being broken down, bound energy is being released, that bond energy is being used to resynthesize the adenosine triphosphate. Okay, so the main molecule that is preferred in cellular respiration is glucose. Other things can be burned in cellular respiration. We'll get to that a little bit later. But this is being broken back down to carbon dioxide and water, and the energy is captured as ATP. Okay. Now, these consist of also two membranes. So, like the nuclear envelope, it's two membranes. However, their nature is quite different. The outer membrane is relatively smooth, but the inner membrane is highly folded. Okay, it has all these folds on the inside. These folds are called cristae. Word should be up there. Yeah, cristae, and that creates actually two important spaces, fluid-filled spaces. Between the two membranes is intermembrane space, and when we get to cellular respiration, you'll see that does have an important function. And then inside of the intermembrane, that fluid-filled space is called matrix. Okay, so when we get to cellular respiration, we'll then spread out on this. All right. Okay. Another energy converting organelle are chloroplasts. Chloroplasts are in plants and algae. It's a site of photosynthesis. All eukaryotes, most eukaryotes, including plants and algae, do have mitochondria. And some of the few protozoans that don't have mitochondria, we know that they came from an ancestry that did have mitochondria, because they have genes for mitochondrial proteins that are located in their nucleus. And how can they possibly be having genes for mitochondrial proteins if they didn't have mitochondria at some time in their ancient past? But this here is only in plants and algae. It is the site of photosynthesis, where photosynthesis is just metabolically somewhat different, the opposite of cellular respiration. When we get to these two things, we'll see that there's a lot of differences between them. They're not just reverse of each other. So this is where carbon dioxide, water, and solar energy has to be put in to make glucose. Glucose is the major product here. Okay, now this also has two membranes, plus an extra membrane. So it has a smooth outer membrane and also a smooth inner membrane. But then it has these stacks of membranous bags that are called thylakoids that are stacked on top of each other, sort of lozenge shape, disc shape. And so we have several fluid-filled spaces here. We have an inner membrane space, a thylakoid space, okay, inside of the thylakoids, and between the thylakoids and the innermost membrane, we have a space that's called stroma. Now, these thylakoid space, thylakoid membrane, and stroma, they're going to these are compartments that will play very specific, important roles when we get to talk about photosynthesis. Okay. Now, what's interesting about these is that both of these mitochondria and chloroplasts, that they contain DNA. Okay, so not all the DNA is in the nucleus. Okay, but it isn't. That's not a big deal. If DNA is there, DNA is there. What the big deal is what is the condition of that DNA? It is not complex with proteins. It's circular. Okay, it's in a single. Sometimes I have duplicate copies, but basically one. What does that remind you of? Bacteria. Okay, prokaryotes. Additionally, they contain ribosomes. Their own ribosomes. Okay? And these ribosomes are not the same as the ribosomes out in the cytosol. They are smaller. They are sensitive to tetracycline and streptomycin. Okay? Furthermore, do I have it up there? Yeah. And they have two membranes, an outer membrane, inner membrane. The outer membrane has beta-beta structures called porins. What do you think all this resembles? Bacteria. Not just bacteria, gram-negative bacteria. Gram-negative bacteria have an outer membrane and a plasma membrane. And so way back in the early 1970s or even late 1960s, I think someone, I forgot his name, came up with the idea that these things were once bacteria. Okay? This is the endosymbiotic origin. And so within the symbiotic origin theory that talks about here is that these things started as in a mutualistic relationship with ancient early eukaryotes. And we believe, and all the evidence sort of supports this, that the first one was for the mitochondria. The chloroplast came later, okay? So if we have some bacterium that happened to get into the early eukaryotic cell, and they live together, they don't eat it, they keep it there, especially if the bacterium could produce lots of ATP compared to the eukaryotic cell, which is what we believe is the case. And they could be eventually, it's a mutualistic relationship. You scratch my back, I scratch yours. And so the bacteria were given a home, they're given nutrients, and what the bigger cell got was ATP. Over evolutionary time, a lot of the genome, a lot of the genes from the bacteria have been transferred to the nucleus. Okay? Mitochondria have well over 400 proteins, different proteins. They can only make about 15 of their own. All the rest of them are shipped in from the outside because they have been transferred, that information has been transferred to the nucleus. They can no longer live outside of the cell. They have lost their identity as bacteria. They are now organelles. Okay? So this is what we believe for both chloroplasts and chloroplast and mitochondria. Yeah, usually I say mitochondria, chloroplast, but I get it the other way around, that means stuff. Okay? All right. This is where I'm going to stop for now. And I'll continue this on Tuesday. So our quiz will cover the material from structural polysaccharides, proteins, nucleic acids, cell structure, all the way up here. Okay?